We recently published a couple of papers. One was a follow-up on some earlier work that I'd done at Emory University and Georgia Tech with Doctors Donald Stein and Michelle Laplaca. And in that study, we looked at the kind of therapeutic window, putting the cells in. Interestingly enough, there's another paper that has come out at the same time by a Dr. Dong's son, and she actually transplanted the cells at the same time points that we did. So we looked at 2 days post-injury, 7 days post-injury, 14 days post-injury, because you want to know, how far out can you go, when can you see an effect. For us, our best time point was 7 days post-injury. For them, their best time point was 2 days post-injury in terms of survival of the cells in the brain after transplantation. So why the difference? They actually transplanted their cells in a completely different area of the brain than we did. And of course, we had 2 different animal models. There's a lot of similarities, but I think that when you that the location of the transplant can be very key, because it has to receive signals from the injured environment in order for the cells to home in. And I know I'm getting off topic here. Anyways, both of these papers that we had just recently published 2 papers on stem cell research, one of which was with animal cells, one of which was with human cells. And in both of these papers, just like all the other papers that I was referring to, the bottom line was that the recovery that we saw, which was substantial, it was significant, was not due to cellular replacement. We didn't see the cells integrating into the brain in terms of, we didn't see cells differentiating into neurons that would replace the lost neurons due to the injury. And the functional recovery occurred too rapidly to be attributed to that in any case. So what we're doing now is we're taking the media that the cells are cultured in, and specifically the media that we're working with is from the human amniotic cells. So it's human amniotic progenitor cell culture medium, we call it ACS. And we're infusing that into animals using an Alzette osmotic mini pump. So it's a little tiny pump that we implant into the back of an animal, and it has a little tube that we insert into the cerebrum, and over a 2-week period, we just continuously pump these trophic factors that are secreted by the cells into the animal. Got very nice beneficial effects just from infusing the animals with the culture media that the stem cells had been cultured in. And we, prior to doing this, we showed that we could keep the viability and the bioavailability of the culture intact at body temperature, by different experiments. So, that's kind of proof in principle that it's not about, right now at least, and I'm not saying that cellular replacement couldn't be an avenue in the future, but right now the bulk of the recovery that we're seeing appears to come predominantly from the beneficial factors that these cells are spitting out, putting out. Well, I hope these kind of results lead to more research on stem cells and traumatic brain injury, and the therapeutic value, I think, is there. I think this field has not been thoroughly explored, and that there is a lot of very, very important work to be done. And so, one would hope very much that it would spark some interest and kind of spur on a stem cell revival, per se, in this area of research. But to do it in a very thoughtful, conservative way. And I think for that to happen, it really, what we need is to get some of the people together that are interested in doing this, almost form a small consortium. Too often we go off into our own labs after a conference like this and we just kind of get an idea and we go with it. But I think there's a real strength when you can gather scientists together and they can share ideas and not worry so much about who's going to get credit for what, but just really share information freely and share the ideas and be respectful of each other's contributions. And you can really make some powerful advances that way. And that's what I'm hoping we can do with stem cell research as it applies to traumatic brain injury in the future.